Hello everybody. Today I'm happy to provide you with this case study entitled Whole Genome Sequencing. AnnotSV revealed a recurrent Alu mediated tandem duplication in IFT140. This video is based on data extracted from the following publication, Whole Genome Sequencing in Patients with Ciliopathies, and covers a novel recurrent tandem duplication in IFT140 that was published in 2018 in Human Mutation. So, I'm going to talk about a family which is illustrated here with this pedigree. There are two affected children born in 1998 and 1999 with a phenotype of ciliopathy. Both children are affected with retinitis pigmentosa, renal anomalies, hand and feet brachydactyly, bone anomalies with a short stature, and only for the first child, a last clinical sign is the overweight. Of course, these phenotypes can be standardized with HPO terms in order to be used in bioinformatics analysis, and you can see these terms here and here. And with that, a whole genome sequencing was performed. So just to describe you quickly what was done in terms of bioinformatic analysis, actually the exome was extracted from the genome sequencing with large overlaps. SNV and small indels were called to annotate, and structural variations were also detected and of course annotated with AnnotSV. Here you can see the summary of the different steps of filtering and analysis. The affected patients are here marked with a star. So with whole genome sequencing, there were 5 million of variants for each patient, whose around 1400 deletion and 2000 duplication. Then the exome was extracted with the V5 bed file given by Agilent and extended by 100 base pairs around. So it was leading to 100,000 variants still. And then all the frequent variations were filtered out. There were then almost 10,000 variants. And because the pedigree of the family was not apparently consanguineous, but recessive, authors focused in homozygous and compound heterozygous variations. Several genes was highlighted, which are listed here. And in particular, a SNV associated with a structural variation in the IFT140 gene were highlighted. Indeed, this gene is implicated in a variety of phenotypes ranging from isolated retinitis pigmentosa to more syndromic cases. Okay, so now let's have a look at this duplication on the AnnotSV web server. Okay, so I can enter the duplication coordinates, choose a good genome build, and set the HPO terms describing the phenotype of my patient. And so now I can submit the job. So here is the waiting page with the ID of my run here. And so I need to wait a few minutes and to refresh this page. Okay, so here is the result page, and I choose to directly visualize the data. Great, so now the duplication is shown in not AnnotSV, and as you can see, I'm using the expanded mode. This duplication is ranked as a class 3 by AnnotSV. It means as a variant of unknown significance, but with a really interesting AnnotSV ranking score of 0.6. If I move the mouse over the gene name box, I have access to additional annotations like the disease from Decipher or GeneCC, which are really close to the patient's phenotype, Mainzer-Saldino syndrome, short rib thoracic dysplasia, with or without polydactyly. I also have access to other information like the mode of inheritance, which is recessive and coherent with our scenario. I can have a look at the location of this duplication in the gene. So this duplication is located from intron 26 to intron 30, and this gene is composed of 31 exons in total. And for example, I have other information like the percentage of overlap CDS of 16%. I also have access to OMIM annotation, like the phenotype or the mode of inheritance, or if the gene is morbid or not. In this case, yes, IFT140 is morbid. And here you can see that the exomizer score is really specific to the patient's phenotype because upper than 0.7. There is no regulatory element overlapped, no non-pathogenic or benign SV overlapped. But if you look just here, there are 5 non-pathogenic small variants that are coherent with the clinic of the patients. And something that is really interesting, the breakpoint annotation pop-ups show that the breakpoints overlapped 2 distinct repeated elements from the Alu family, Alu-GR and Alu-GB. So that is really suggestive of a recurrent genomic mechanism. Okay, so these 2 Alu repeats identified in the breakpoint junctions have a microhomology of 6 base pairs, suggesting that the duplication was mediated by Alu-Alu recombination, and suggesting that this duplication could be recurrent and happen many times. So why not to see whether other patients can be identified with this tandem duplication? So the lab from this publication contacted collaborators, and a large screening of several cohorts was carried out. Authors were successful with 11 novel families, and to conclude, the recurrence of the tandem duplication in IFT140 was revealed thanks to the AnnotSV annotation. Well, that's it. If you have some interesting data to share publicly in a video, please let me know. I always do care, and it could be useful for the AnnotSV community. Bye bye!